All right, good afternoon. We're doing another journal club, and I actually want to give you an introduction to this journal club that is going to give you the biological side of things. And then we're going to go to the technological side of things, and you'll see the differences in how antibodies are made in the normal biological mammalian system and in a device called the antibot. And so that's our main question we're going to start with. How does your immune system make so many antibodies? Now, we've mentioned some of this before. This takes part in the life cycle of the B cell or the T cell. And we're going to focus on B cells here. The same process takes place with generating all of the different T cell receptors that we have. It's just a little less complicated, but the same shuffling goes on. And we're going to focus on the development in the bone marrow or the thymus where the BCR and TCR are generated, where you have the cell growing up and it's going from basically not even a B cell or a T cell at all to becoming a naive one, which has one copy of its receptor. So you also have a step where you have to eliminate self-reactive cells. That's a really cool part of immunology, but we don't talk about that yet. We'll talk about that when we talk more about the thymus and autoimmunity later. And so you have to have an activation. Remember that they are made, they get a particular set of genes that will make the BCR or the TCR, and they only make that version. Then they are activated either in the lymph node or the spleen. They differentiate into effector cells, and they can also differentiate into memory cells as well. So their function as an effector cell are all the things we talk about, their jobs to do. They can be T helpers, they can be T killers, or they can be B cells that produce antibodies on their own, and with a little help from the T helper can become a plasma that is a real turbo producer of antibodies. Now, remember that for all of these will undergo apoptosis relatively quickly, a matter of weeks, I believe, except for a few that stick around and become memory cells. Just remember, apoptosis is a natural part of the cycle of life. When you have an infection, you want a lot of that particular B cell or T cell. When the infection is gone, then you actually don't need it, and you don't want to spend resources to keep those cells alive. You do keep a few of them alive as memory cells, and that's how you can remember a vaccine that you've had 10 years ago. So we have this slide that shows you that there are different cards in the deck that could be drawn. And I want to go into a little more detail because this slide is very vague about how the cards are shuffled. And it's actually really kind of a cool technique that combines the best of upper division biology. It's a cell biology and biochemistry working together. So it's very much a topic for this class. So the observation is that we have a lot of different antibodies. There's a lot of diversity. And McFarlane was right that you have a ton of diversity, but each cell only does one thing. Each cell only makes one receptor. So the puzzle is, how do we have so many diverse gene sequences for exactly this one part of the genome when the rest of it stays very stable? And when we look at antibodies, we can see a part of the answer. There is a variable region that changes a lot more than the constant region. And the variable region is where the diversity is generated. So we can already see there's part of the gene that the cell focuses on to increase its diversity while the rest of it stays the same. So the experiment was to take embryonic liver DNA, which would be the germline. This hasn't differentiated at all. And then take a B cell DNA. It's easier to get a lot of DNA from a tumor. And so a B cell tumor DNA was taken and sequenced and compared to the same DNA in an unrelated organ. And it showed that the DNA in the B cells was always rearranged. In the lymphocyte, the V and C segments were put together. If you look at the liver, it's got a bunch of V segments and a bunch of C segments. In the B cell, it doesn't have a bunch. It's got one and one. So the other ones have been cut out, and you've taken two of the segments and you've put them together. So with more sequencing, eventually realized that the V and C segments were only the biggest and easiest to see. There are also smaller segments that are J between the V and C, so they're joining segments. And then there's also in the heavy chains, they even have a fourth kind of segment, which is D, and they ran out of names, so they just said these generate diversity. Let's call them diversity segments. And so germline DNA was going to look like a bunch of V segments with a C segment and maybe fewer C segments, and maybe just one C segment in some cases. That's all you need because you want it to be constant. If you have cells that can switch between different types that have different Cs, I think you will see different Cs there. But the main thing is variable, you have a lot of them, and the B cell DNA, you have the variable, you have just a few of the variable regions that are, the other ones are snipped out. And so how could this evolve? And we do want to always ask the question about evolution. Natural selection could work with this system because it's basically natural selection on a cell biology level. You have to generate the variation, and then you don't have a problem of selection. Selection is that you bind to the antigen and that you eliminate the infection. So the only question is, how can you generate the variation, not how can you select within that variation? And you want a lot of variation because you want to be able to recognize anything that pathogens can try to come up with. They're trying to come up with new structures that will evade immunity. So you need some kind of way to recognize those not previously encountered, and the solution for that is to have a huge library of receptors sitting around, and at least one of them will bind at least kind of well, and then you actually have a separate sort of program that actually hones and increases the binding strength of the one that you've selected. So it's really cool. But you've got to realize also, if you have a bunch of receptors that can bind to anything, you don't want them to bind to yourself. You don't want them to bind to self proteins. And so there are education programs, especially for the T cells. The T cells have education programs, the thymus, what do you call it, the murder factory or whatever it was. But basically, you delete, apoptosis is my preferred term, you delete the self-reactive cells, or at least you should. If something goes wrong with that, and you have a few self-reactive cells get through, that's the basis of autoimmunity. So just remember, receptor diversity is there from the beginning. And then when an antigen is encountered, it's not a measure of making a new sequence, but of selecting an existing sequence from a huge library. And so, because this is a cell biology talk, essentially, there's a little bit of cell biology from Bio 2000 level that we have to remind ourselves of. Just remember, when we're talking about a gene, we're talking about the DNA. And DNA is usually very stable. In B cells, we have a special set of genes that actually makes it unstable in a way that generates diversity. Transcription is the process of turning the DNA into an RNA transcript, and then that transcript is spliced together. And actually, you can generate diversity at either the DNA level or at the RNA level. Then you have a mature mRNA after it's been spliced and it's ready to be turned into a protein. Protein is a different language. You have 20 amino acids rather than 4 nucleotides. You have to go through a translation process, and it's a very different kind of molecule. So you make the linear polypeptide that corresponds to the linear mRNA, and then that polypeptide has to fold up into a 3D structure. This process of folding and assembly and maybe some parts being cut out, proteolized out, that is going to give you the mature protein. So you have immature and mature RNA, and you have immature and mature proteins at this level as well. I guess in a sense you have immature and mature DNA because you have the DNA of the liver that doesn't have any VC combination, and then you have the B cell DNA which has been recombined. So the combinatorial diversity, just remember we have this picture of what antibodies look like. You have 12 domains, you have light chain, heavy chain, variables are at the end, and the other 8 are going to be constant domains. And so the light chain doesn't need as long of a sequence, and the light chain also has fewer gene segments that it's made from. Remember that we mentioned V and C were found first, and you found J, and then they found D for only heavy chain. So just remember that light chain is made from 3 gene segments, and heavy chain, being longer, has a 4th segment that is shuffled into the mix. Oh, and by the way, look down here at the bottom. There are 2 kinds of light chain. There's kappa chain and there's lambda chain. Then there's heavy chain. We have actually 5 different kinds. Usually we don't focus on that too much because the light chain lets us see how there can be two different kinds of light chains. That is the only thing for you to notice. There are different kinds of constant domains, but those can vary by cell type and things like that. Here's a picture of what the DNA looks like. This would be, the top is what the liver cell light chain DNA would look like, but the second one down is what it would look like in a B cell. You have 3 V kappas, you have 4 J kappas, and you have 1 C kappa. In the B cell, you see that one of each has been selected and then joined together with a little extra sequence. We're just talking about the basics of what goes on. There's actually a few other steps that add extra diversity into this. A very cool process. So for the heavy chain, you have the same thing going on, except you have all these D segments in the middle as well. And it turns out that you actually pick 1 V, 1 D, and 2 J's, along with the C, whatever the Greek letter is, for that type of heavy chain. You see how it's the same basic idea, but the heavy chain just has more segments that it can be built from, partially because it's longer. And look how short these are, by the way. You can see that the V segment is about 100 amino acids long. The D segment ends up encoding only a couple of amino acids. It's usually amino acids 102 to 106 in the final antibody would have been contributed by the D segment. Then you have a longer J segment, but even that is only less than 20 amino acids long. So the V segment is by far the largest, but you do have these different possibilities joined together. And so here's an example of the kappa chain DNA, and you can see that we have the exact number of kilobases between them. They've worked out exactly where the gene segments are and where the spacers between them are. And really, you can just trust that a lot of this biology works. There's a little bit of mixing up sometimes. We don't want to say that all of the V's are always together. It looks like on the lambda, you actually have 1 V lambda that's all the way over here. It looks like there's a little bit of shuffling that's gone on in evolution that's been sort of baked in. But it's pretty close to being V, D, J, and C in order. And so that means that this is one of the few places where we have a calculation in immunology. It's really a basic probability type of calculation. If you have X number of choices for one thing and then Y number of choices for another thing, then your total number of choices is X times Y. So if you're adding segments, you're multiplying the possibilities together. So for example, if there's 45 heavy chain segments in V, and there's 23 possible D segments, and then there's 6 possible J segments, well, if you just have 1 V, 1 D, and 1 G, like J, like you do in most heavy chains, then it's 45 times 23 times 6. Even just that is 6,210 different, and that's just for the heavy chain. That can be multiplied if you add it to a number of light chains. So for example, if you see the number of kappa chain segments, they don't have the D's in place, but they have 41 times 5. And just that is 205 for the kappas, 33 times 5 for the lambdas, and then you actually look at all those together. So the number of heavy and light chain combinations, just from the gene shuffling, is going to be 205 plus 165. So you only pick one of the either kappa or lambda, but that's still about, what is it? I can do it in my head. 370. I think I did that right. Okay. So 6210 times 370, and that ends up being 2.3 hundred-thousand possibilities just from this normal kind of gene segment combination. Now, in reality, some segments are used more frequently. We actually have a study on that at the very end. The combinations don't appear to be totally random, but for a undergraduate level class, we can make you calculate what it would be if they were completely random and realize that real life is a little more complicated. So this diversity is generated by taking genes and rearranging them. Now, if you've had enough biochemistry, you might have seen an enzyme called a transposon, or a recombination type of enzyme. Recombination is the process, and actually it's talked about in cell biology as well, where DNA is switched around. And this is one of the main places where we see recombination, because this is where the cell wants to switch DNA around. Most of your DNA, you want to keep it stable, but you do have enzymes that switch it around if you need to. And these enzymes work on particular gene sequences. We can see exactly how they work, exactly where they like to bind, where they like to cut, and where they like to move stuff around. And so it's literally cut and pasting it together, but that's inherent in the DNA possibility, because the phosphate backbone is inherently the kind of thing that you can break apart, you can hydrolyze it, and then you can rejoin it together through the attack of the phosphate onto the 3' phosphate, the OH attacking the phosphate. Okay, that's a little more biochemical structure than we need for this class, but if you do remember it, this is where it comes into play. Okay, so the segments of the DNA can be pasted together, and they can construct. Remember that there are also two chains for TCRs. There's an alpha and beta chain, and they're symmetric. They're just 4 domains. So they're just like one arm of an antibody, but it's the exact same principle. They have different segments that are pasted together, and alpha and beta form a dimer that forms the complete T-cell receptor. 2 domains each, 4 domains total. There's also, even a little more complicated, there's a different kind of T-cell receptor in the gut, and it's called gamma delta. But again, the domain at this level, the domains look exactly the same. So these are all very similar. To do this, you need a special enzyme called a recombinase. And this is particularly named the RAG1 or RAG2 recombinase. And you also need a few other things. I'm not going to focus on the other things that you need. I want you to focus on the recombinase. Just in case you're interested, there's other things called the terminal transferase. There's some DNA repair enzymes, so that unit in biochemistry is actually relevant here. But we don't require that for this class, so I'm just going to leave it at recombinase. And the recombinase works at recombination sites, which have recombination signal sequences at one or both ends. And so we know exactly what that DNA is that the recombinase will bind to, then it will cut it, and it will paste it together with another site. Very, very cool how that works. Now, after this, that's all you need to make a TCR. To make a BCR, you actually want to sort of make it a little bit, you want to tune it to be a little bit better. And there are a few things that happen. There's another kind of recombination called class switching. But the thing that's most important to me is there is something called somatic hypermutation, which is where individual amino acids are hypermutated at points where they will make the most contact with antigen. This is like re-editing your draft paragraph that you've written to take out the clunky punctuation and to put in a really nice sentence. It's small changes, but focus changes. And it's just amazing to me that the immune system has evolved to do this. So these RSSs are where the recombinase works. And here they are. This is exactly where they are. We can even put this into DNA, and if we can get the recombinase working, it will do the recombination for us. It will take these sequences. If you have this sequence on the left and this sequence on the right, then it will actually bind to both of those sequences, and it will join the V on the left with the J on the right and bring them together in the final. There are some rules about how it has to work. There's a certain sequence that has to be left behind. You can't just do this. It doesn't leave without leaving a mark because of the nature of the sites. It leaves like a half site behind once it leaves. And you can see that. So we can see that in germline DNA. And actually, I say that we can see these in the germline DNA before they are joined together. I might have misspoken right there. Let's leave it at the germline for right now, and let's see what else we get. So the light chain recombination will it will have these spacers that are originally there. The spacer will be removed. And so what I said just a second ago, that it leaves a mark behind, actually it looks like it doesn't really. It always works at the same kind of place, but the spacers are removed when you actually join together. Look at how the yellow spacer on the left and the brown spacer on the right. Segment 3 of V is joined together with segment 3J, and you end up having the rearrangement happen. All the stuff in between is just cut out because it's not going to use it, and so you see that those are missing when you look at the B cell because it doesn't care. It just cares about putting together the right V or the random V with the random J. There are, so the other segments that are left behind, actually those are removed at the mRNA level. There is some sequence removed at the mRNA level, but the V and J's are joined together at the DNA level. So there's also a rearrangement that puts together V, D, and J. And same kind of thing. You have spacers that are at specific places, specific sequences. You end up taking one blue, one purple, and one red, and you join it together. It's called VD rearrangement and DJ rearrangement. I think that's kind of obvious if you know what V, D, and J are in the first place, which is the thing I want you to know. And so just think about this. One genome can give rise to a completely different genome. Now this is just in one part of the genome in a very focused way, mediated by a very specific enzyme. So this is tightly controlled, but it does show the principle that DNA, the mutations in DNA can be sort of sped up and directed by the cell for a purpose. Evolution is not purposeless, at least not at every single level. And when you look at it, you can see where this purpose comes from. And I think that's one of the fascinating things we can talk about in seminar and capstone courses. In immunology, we don't have time, but I can set you up for later. And Siddhartha Mukherjee has a good example of this in his great book, The Gene, which really helps to put things together. So he says, like, if you have any sequence of letters, and DNA is like a sequence of letters, you can reshuffle it to get an entirely new sequence. Usually it won't make sense if you take letters and just shuffle them, but sometimes it can make sense. So let's say that you have this thing that spells out structure and genome. And if you just take the C and the T and then the OME from genome, the C and the T from structure and the OME from genome, and you shuffle them together in this way, if you can only use one sequence from each kind and you're trying to use, you can use two sequences from the left, from the red, you can use one sequence from the right, and you can only use one, once you use one, you can't use it again. If you have those rules, you can calculate exactly how many possible words you can make. And again, most of them will be nonsense, but by these rules, you have 5 units in structure, and you're picking 2. You have 3 units in genome, and you're picking 1. So therefore, you have 5 possibilities times 4 possibilities times 3 possibilities. I can do that. And actually, you know what? I actually miscalculated that right there. Here, let me fix it. Here we have it times 3. It's 5 times 4 times 3. So I did the calculation right. I just wrote down the thing wrong. Let me bring it back. There we go. All right. And let me just do one thing. Hopefully I won't confuse the computer too much. I want to make sure it's still hearing me. Oh, good. It's still hearing me. That's nice. Good computer. All right. And this word makes sense. He likes, it's really reshuffling, and if you reshuffled all 60 possible words, then you would end up with, reading down the list, see a lot of gobbledygook, but then you would see, oh, comet. And comet actually comes from structure, genome, reshuffled. So you can get a lot of diversity just from that kind of simple example. And this is the enzyme that does it. We know the exact structure. It's very cool how it works, but again, you just need to know the name. You don't need to know the biochemistry for this class. It cuts DNA, it makes little hairpins out of them, and it's only expressed at a certain point. It's only expressed when the cell needs it, and only needs it when it's first developing. So once it's a naive, grown-up B cell, or even childish B cell perhaps, once it gets to that stage, it doesn't express this anymore because it doesn't need to reshuffle. You reshuffle once at the beginning of your life, and then you keep those for the rest of your life. Okay, so, and it looks like a transposon. We have bacterial transposons that shuffle DNA around just like that, and yet this is a mammalian immune system protein that has to do with human diseases. Well, it looks like we borrowed their notes. And just remember that, if you look at different B cell sequences, this is looking at a certain kind of B cell within, actually this is looking at all sorts of B cells that you line up. Within the sequence, so you have, this is the light chain V region, so it's only about 100 residues. You go residue by residue and you sequence a bunch of B cells and you say, what are the residues of antibodies in order? And you'll always see a lot of variability at particular points. You'll always see a lot of variability around residue 30, around residue 50, and around residue 95. And you see that those stand out on a graph. We color those red, and those always have higher variability than the things around them. If you think about that, this variability could not come just from gene shuffling. There must be something else going on, and there must be a way that the cell actually targets these. It's called somatic hypermutation, because it happens to somatic cells, and it's hypermutation. It's not that bad. Detmer seems scared of it, but you can handle it. And so you have this somatic hypermutation that increases the variability at these particular, like 10 amino acid sequences, and they're called highly variable regions. In the V region, you have 3 of those, and if you map them out, they don't map out onto the beta strands or the middle of the protein, they map out into the very tips. And so we call those, they must be there to increase the fit of the antibody to the antigen. Now, if you look at these, they also have 2 names. They're called HV3. Technically, when you're talking about the gene, you're talking about HV3. When you're talking about the protein, you're talking about CDRs, complementarity determining region. They're both the same thing because the highly variable regions are the same things that determine the complementarity of antigen to antibody. So there you have them. The antigen binding site is made up of 3 loops in the light chain and 3 loops parallel on the heavy chain. On each thing, they come together and they form one antigen binding site. And there you get your 6, and they are deliberately mutated to be more variant than even gene shuffling would say. And you've seen something like this before, because we've seen the same variability chart. It's not quite as stark as it is with antibodies, but you see the same thing with MHC. Remember how the platform underneath the pockets, you had a lot of mutation, particularly where the MHC touched the peptide, the anchor residues in particular. So we've shown you this before. That was with MHC. Same thing with antibodies, but each of the variable regions are always where they need to be for the antibody or for the MHC to work best to bind a lot of different things. And so the question is it really random how VDs and Js go together or not? And they actually have just investigated. This is a little tricky to investigate because you've got to sequence a lot of genomes, basically, to get an idea of whether it's random or not. You also have to sequence a lot of different antibodies, which requires like single cell type sequencing. You collect circulating B cells, and then you sequence each one of them. Now, the library of naive antibody sequences that you have sitting around in your body, in a B cell somewhere in your body, is at least 10 to the 12th. And you only have 5 billion blood cells. That's 5 times 10 to the 9th blood cells in circulation that are B cells. That means only a fraction of what could be circulating is actually circulating. So the question is, are these 5 billion, are they just randomly selected from the 10 to the 12th that you could have? Or are there trends? Is there a logic behind which antibodies are circulating in your body? And is there a way in which naive antibodies are sort of biased in one way or another? And the take-home thing is that they are not completely random. They did find some patterns there are some B cells that show up more than randomness would predict. Okay? And so just look at this. This required almost 3 billion antibody heavy chain sequences, and they just were looking at heavy chain. That's why it took a long time to come out. This happened in the last 10 years, I believe. And just look at this figure from the paper. What they did is they sequenced V, D, J, and C regions for the heavy chain. And so it's just what I showed you. They just made primers, and they did, the primers are required for the PCR, and they did a lot of different kinds of PCR type sequencing. They actually did advanced sequencing rather than old school PCR, but the idea is the same, that you read off the DNA sequence this way. And by the way, it's Illumina sequencers. They're a big name when you're ever looking at doing this kind of work. And I know there's lots of jobs in the industry for doing Illumina sequencing of your cancer cells so that you can see exactly what's in your cancer cells, for example. They did this with B cells and only for the heavy chain region to look at VDs, Js, and Cs. And so a particular combination of VDJs and Cs are going to be given a different number on this graph. Well, first let's look at the top of the graph. The top of the graph, each person is a different color. So it's a very pretty graph. And it has, so one person was red and a different person was orange, a different person was yellow, you know, all the way down. This just shows, what is it, 9, 10 different people? Yeah, I can count. 10 different people. So they just had 10 different people, but they sequenced all of their heavy chains, and they found out, for example, that most people have about 80% are IgM receptors. Remember that we said that most naive B cells are IgM before they grow up and become IgG? And about 20% are IgGs, but there's a little bit of variation. Each person is a dot on the point up there, and you can see that it's exactly symmetric. The IgGs plus IgM frequencies add up to be 1. And you can see there's a little variation, but everyone always has more IgMs than they have IgGs. Okay. And then you look up and down, and there are some that are faint. That means that they don't have that much of that particular VDJHC combination. CH, sorry. But you see that this one I circled right here has dark colors. That means that they saw a lot of it in everybody. This one over here is a J gene, and this is actually separated out by V gene and J gene use by subject. And there were certain sequences, like whatever was given this, there's a particular V gene that everyone uses. There's a particular J gene that everyone uses. The rest of them are relatively distributed evenly so that everyone has a different mix of those. So that means there are some gene segments that are used more than others. If it was completely random, they should all be the same color, the same intensity of color. But some are faded, and some are, in everyone, some of them are dark colors. So the immune systems do have some favorite gene sequences. We'd call those clonotypes that are shared among different people. But these come from, they tested these, and they wanted to see, are these because there are particular sequences that bind to antigens better? Are there like antibody sequences that are kind of like toll-like receptors or innate immunity? You make one because it binds antigens better? The answer is no. It looks like these come from the way B cells develop, rather than some kind of choosiness that comes from the developmental side, not from antigen binding requirements. So the diversity available is usually in the region of 10 to the 16th, 10 to the 18th. 10 to the 12th, by the way, is a minimum. And despite this, there are some that you see that you would more than expected. And they found that it's probably driven by the selection processes that are involved in B cells developing in the first place, rather than them converging on binding a possible antigen. I think that's kind of cool. It's just kind of, and this might have some therapeutic use, but to me, it's interesting in teaching immunology. And we want to think about what that says. So there is a convergence in antibody evolution, although it doesn't have to do with antigen binding, but there are ways that antibodies have to be made. And we all make some more of these antibodies that just happen to be more useful to be made, or more likely to happen because of the way antibodies are made. So evolution is not completely random. And I just think this is really interesting, because if it's not completely random, maybe there's a selection for the not completely random parts of it that will allow for better survival. And maybe there's a reason for it. This is one of the great things that biology can do. Rather than shows that some parts of life are driven by randomness, but some parts of life are not. And perhaps some parts of life are selected from a random pool, and that still means that they are selected for a reason. Selection itself is a sort of imposition of order. So evolution is not all about disorder and chaos. It's about an interplay between order and chaos. And antibody evolution might have something to say about this. Here's another paper that would be interesting if somebody wants to look into it. If you ever want to talk about an evolution paper, you can say, well, what can I talk about antibody evolution? It's different than Darwinian evolution. It's directed and designed in a certain way. But it does have some things where, if you look right here, there is an intrinsic mutability of a particular VH gene. which closely resembles another VH gene. In other words, there is something that evolution does repeatedly, and it's not just completely random and statistically even. There is a bias, and it's kind of nice to know that there are things for science to find and for us to figure out. If it was completely random, there'd be nothing to figure out. But I actually find it to be, it's one of the things that I like looking at as a theist and as a Christian, to say, if God made an orderly universe, then God put us in this. He didn't tell us everything about how antibodies work, but he gave us the brains and tools and hands and an orderly universe to be able to figure it out. So when you hear evolution, yeah, you can think parts of it are driven by randomness, just like parts of gas diffusion are driven by randomness. That does not mean that it is purposeless. And I wrote a whole book about this. So, enough about that. Let's talk about the rest of things in a second.